When I was 22, I was diagnosed with stage 4 melanoma. None of the approved treatments at the time worked. I thought that my life was over, and it hadn't even begun yet. And then, a miracle happened.
Sharon Belbin was the first patient that I met. It became clear to me that this basic science that I had been working on was saving lives. And at that time, surviving advanced metastatic melanoma was something that was essentially unheard of. There was this aha moment, where the tumors just melt. Virtually no one thought that immunotherapy would work until it finally did.
Immunotherapy turns on your immune system and allows your body to then fight cancer.
The answer to cancer may already be inside every one of us. Thanks to decades of research, scientists are now learning how to unlock the power of our own immune system to treat many different types of cancer.
There's a unique window of opportunity here to really understand how these different immunotherapeutic drugs work together and with other agents to benefit as many patients as possible.
Saving lives starts with the research, and getting cures for all cancers means going beyond the science we already understand.
The Cancer Research Institute has been the primary organization that's been supporting the notion of treating cancer with immunotherapy, and knows better than any other organization how to get in touch with the right people, fund the right kind of research, and move this new therapy to even better heights of successfully treating patients.
Funding from the Cancer Research Institute validates our ideas and gives us confidence to pursue them.
A revolution in cancer treatment has begun. Immunotherapy is saving lives today and has the power to save many more in the future. The Cancer Research Institute sees a path to lasting cures and is committed to funding the most promising science wherever it's being done.
It's not just a scientific puzzle.
Immunotherapy, it means hope.
I want every person to have the same chance at life that I had. I know that the Cancer Research Institute won't stop until that becomes a reality for every cancer patient.
Hello, and welcome to the Cancer Research Institute's 35th Annual Awards Ceremony and Future Look Roundtable. I'm Dr. Jill O'Donnell-Tormey, CEO and Director of Scientific Affairs at the Cancer Research Institute. I'm delighted that you have joined us in celebrating one of today's most important medical innovations, mRNA vaccines. Today, we will honor 4 scientists whose groundbreaking research and tenacious belief in the technology they developed not only has made possible life-saving mRNA vaccines that protect against severe cases of COVID-19, but also has opened the way to new and exciting approaches to cancer treatment. The bridge connecting these two very different threats, COVID-19 and cancer, is immunology. Immunology is the field of science that studies the immune system and seeks to understand how it works and how it can be harnessed to prevent and treat a vast number of diseases. The Cancer Research Institute is the first nonprofit organization to explore the immune system as an answer to cancer, and has focused on this goal since our founding in 1953. This work over the past 7 decades has made possible today a revolutionary new way to treat cancer called immunotherapy. As we'll learn in a short while, immunology research aimed at curing cancer allowed a rapid pivot of mRNA vaccine technology toward targeting COVID. COVID vaccines have made it possible for us once again to safely visit friends and family, dine in restaurants, travel, and so much more that was paused indefinitely when the pandemic first struck us nearly 2 years ago. As we return to a new kind of normal, thanks to these vaccines, it's hard to believe that it's only been 1 year, almost to the date, since an FDA panel reviewed the first evidence that this approach was highly effective, bringing much needed hope and relief to many of us and preventing countless deaths. This is the power of science that saves lives. The Cancer Research Institute has always been committed to funding the most promising scientists and supporting research with the greatest potential to transform cancer treatment with immunotherapy. This vital work is possible only with the generous support of you, our donors and supporters, to whom we say thank you. Whether you give as an individual or on behalf of a corporation or foundation, you are an invaluable partner in pushing scientific research forward to save the lives of more people with cancer. Today, we present the William B. Coley Award for Distinguished Research in Basic Immunology to 4 scientists who embody CRI's core commitment to scientific excellence and who share our belief in the potential for immunotherapy to treat all types of cancer effectively. Here to introduce the Coley Award is the Director of CRI's Scientific Advisory Council, Dr. James Allison. Dr. Allison is Chair of Immunology and Executive Director of the Immunotherapy Platform at the University of Texas MD Anderson Cancer Center. Dr. Allison has received countless awards, including CRI's William B. Coley Award in 2005, and in 2018, the Nobel Prize in Physiology or Medicine for his groundbreaking discovery of immune checkpoint blockade, which led to the first FDA-approved checkpoint inhibitor drug, giving thousands of patients with advanced cancer much-needed new treatment options. Jim's work has revolutionized and energized the field. Welcome, Jim.
Thank you, Jill, for that introduction. It's an honor to join in celebrating this year's 4 honorees whose exceptional contributions to science and medicine have earned them the highest scientific honor from the Cancer Research Institute, the William B. Coley Award for Distinguished Research in Basic and Tumor Immunology. The Coley Award honors the legacy of the late 19th century surgeon who today is regarded as the father of cancer immunotherapy. His pioneering work inspired his daughter, Helen Coley Knotts, to found the Cancer Research Institute nearly 70 years ago, long before anyone believed that immunotherapy would one day be an effective answer to cancer. Since 1975, the CRI has bestowed this award upon 111 of the most influential scientists in the fields of immunology and tumor immunology, whose individual or collective work has made and continues to make a meaningful impact on the treatment of cancer as well as on infectious and autoimmune diseases. These scientific pioneers have unlocked some of the most important mysteries of the immune system, from characterization of the biology and function of its basic components to the discovery and development of new treatments that unleash our immune system's power to fight and even eliminate cancer. Coley Award winners have later gone on to win some of the world's most prominent scientific accolades, including the Lasker Award and the Nobel Prize. In this way, the Coley Award serves as a predictor of impact, given only to those scientists who truly transform the field. This year's honorees are no exception. Presenting the Coley Award this year is Associate Director of the CRI's Scientific Advisory Council, Dr. John Wherry from the University of Pennsylvania. There, Dr. Wherry serves as Director of the Institute of Immunology and is also co-program leader of the Immunobiology program of the Abramson Cancer Center. Dr. Wherry's lab is at the forefront of understanding how chronic infection and cancer can affect the function of the immune system's T cells, and is also driving new insights into how other types of T cells help facilitate protective antibody responses following vaccination, which makes him an excellent choice for presenting to our honorees this year. It's my pleasure now to welcome Dr. John Wherry.
Thank you, Jim. At this time last year, the world was gripped by the COVID-19 pandemic with no end in sight. The battle isn't over, but thanks in large part to this year's 4 Coley Award honorees, Katalin Karikó, Drew Weissman, Ugur Sahin, and Özlem Türeci, we are now armed with vaccines that are exceptionally effective at protecting people from the coronavirus SARS-CoV-2. Collectively, their decades of pioneering research into messenger RNA, or mRNA, not only led to the 2 highly effective COVID-19 vaccines, but also opened up entirely new ways of addressing a variety of diseases, including cancer, HIV, malaria, and sickle cell anemia. The genes in our DNA contain the blueprint for every protein produced in our cells. But first, the information must be sent to the cell's protein-producing factories, and that's where mRNA gets its name, as the messenger that delivers that information. By supplying specific mRNAs to cells, one could theoretically get them to produce any protein imaginable. One could, for instance, use mRNA to cause the body cells to create the coronavirus's spike protein, so that the immune system could be alerted to its presence and develop a protective response against it before a person is exposed to the real virus. Or perhaps use mRNA to temporarily increase the production of a normal protein that a person's body isn't making enough of, such as a growth factor involved in blood vessel formation. While mRNA is very simple and easy to make, successfully getting the mRNA into cells where it can trigger protein production is no simple task, because the immune system sees naked or unmodified mRNA as a potential threat and quickly destroys it before it can do its job. Fortunately, in 2005, Katalin Karikó and Drew Weissman made an important breakthrough showing that you could cloak mRNA in certain molecules in order to sneak them past the immune system and get the mRNA into cells to produce protein. This approach and the technologies arising from it were licensed to both Pfizer-BioNTech and Moderna for COVID-19 vaccines and paved the way for a number of other mRNA-based strategies in medicine. Building upon Karikó and Weissman's work, as well as research and development of their own, Ugur Sahin and Özlem Türeci, the co-founders of BioNTech, picked up the torch of mRNA therapeutics and ran in the direction of cancer. In 2012, they used mRNA to create the first personalized neoantigen vaccines against cancer. And soon after, they recruited Karikó to join them in this endeavor. These efforts provided them with the crucial experience and technological prowess that they would summon later when COVID-19 came knocking so abruptly at the world's door and vaccines were so badly needed. These vaccines have already prevented countless deaths and will prevent many more in the years ahead. Today, mRNA-based vaccines to treat cancer are also showing promise in preventing melanoma recurrence after surgery and are being evaluated against other advanced cancers alone and in combination with checkpoint blockade immunotherapies. Like immunotherapy, many also used to doubt the value of mRNA as a therapeutic. But thanks to these four pioneers, whom we are honoring here today, tenacious individuals who kept on pushing in the face of doubt and followed the science, mRNA's value in medicine and its potential to address global health crises is now undeniable. While mRNA has made its biggest mark so far against COVID-19, its future impact against cancer may be no less great. And in time, mRNA-based therapies may come to redefine the way medicine as a whole works. Therefore, on the behalf of the Cancer Research Institute's Scientific Advisory Council, I am proud to present the 2021 William B. Coley Award for Distinguished Research in Basic Immunology to Drs. Katalin Karikó, Ugur Sahin, Özlem Türeci, and Drew Weissman for their many contributions that have advanced the field of mRNA therapeutics, and in particular, enabled the development of the world's first COVID-19 and personalized cancer vaccines. It's my pleasure to present our first Coley Award to Dr. Katalin Karikó, who is currently a Senior Vice President at BioNTech, as well as an Adjunct Professor of Neurosurgery at the University of Pennsylvania Perelman School of Medicine. Dr. Karikó.
Thanks, John, for this nice introduction. It is a great honor for me to receive the 2021 Coley Award along with my outstanding colleagues, Ugur Sahin, Özlem Türeci, and Drew Weissman. I am so glad that I had the opportunity to work side by side with all of them through the years. This award recognizes not only us, but all of our colleagues with whom we work together. It is also a real privilege to be among the previous Coley Award winners, the most distinguished scientists who laid the foundation of immunology, how we understand today. I began working with RNA as a graduate student in 1978 and enjoyed learning all the exciting characteristics of this fascinating molecule. I learned the way that RNA can be made, modified, how it can be delivered into the cell, and the magic as it instantly translates into protein. I love the enigmatic nature of mRNA, which this year became an approved drug in the form of COVID-19 vaccine. I am thrilled to know that I was one of the many scientists who contributed to the success of mRNA vaccine. Together with my colleague, we inched toward solution, building upon discoveries of scientists who came before us and created an optimal RNA suited for therapy. Never in a million years I would imagine that it would be used to create a vaccine to combat the global pandemic. I would like to say thanks to all of those who helped me in my journey, my family, my teacher, my mentors, my fellow scientists. Thanks to all of you.
Congratulations, Katie. It's now my pleasure to present our next Coley Award to Dr. Drew Weissman, who serves as the Roberts Family Professor in Vaccine Research at the University of Pennsylvania Perelman School of Medicine. Dr. Weissman.
Thank you, John, for that kind introduction. And I want to thank the Cancer Research Institute for honoring us with this award. But you have to realize it isn't us getting this award. It's thousands of researchers before us who did all of the preliminary studies and all of the important work that led Katie and I to discover nucleoside-modified mRNA. And it's the thousands of researchers after us who are going to take what we discovered and use it to develop new therapeutics, vaccines, and other therapies. The research world is a collaborative world. Everybody works together. So while we're getting this reward, everybody needs to be awarded for what's been accomplished because we all work together to perform this and to develop these vaccines. Thank you.
Congratulations, Drew. It's now my pleasure to present our next Coley Award to Dr. Ugur Sahin, co-founder, Chief Executive Officer of BioNTech, a professor at the University Medical Center of Johannes Gutenberg University, Mainz, and the co-founder of the biopharmaceutical nonprofit translational research organization, TRON. Dr. Sahin?
Dear colleagues, I'm deeply honored and humbled to share this award with Özlem and our esteemed colleagues, Katie and Drew. The William Coley Award is special to us. On the one hand, it recognizes research and the medical application, both which are close to our hearts. On the other hand, and perhaps more importantly, so many of our esteemed colleagues, immunologists and cancer researchers, have received this award before us. Colleagues whose achievements and accomplishments we hold in the highest regard. So it is a very special honor to be included in this list of awardees. I would also like to thank our early research team with colleagues who started as Ph.D. students and young postdocs more than 25 years ago and laid the scientific foundation of our mRNA vaccines. Finally, I would like to thank to many colleagues at BioNTech who have supported us in the years since then to further develop the technology and set up the production. And of course, I would like to thank our Lightspeed team and the cooperation partners at Pfizer, among others, with whom we were able to successfully develop this vaccine in such a short time. Thank you again for this wonderful recognition.
Congratulations, Ugur. Lastly, it's my pleasure to present our final Coley Award to Dr. Özlem Türeci, the co-founder and chief medical officer of BioNTech, as well as the president of the Association for Cancer Immunotherapy and chairperson of CI3, Germany's Cluster of Excellence for Individualized Immune Intervention. Dr. Türeci?
Thank you so much, John. Dear ladies and gentlemen, dear colleagues and dear friends, I'd like to thank the Cancer Research Institute from the bottom of my heart for recognizing our work. I'm deeply honored, feel humbled and overjoyed to receive the William B. Coley Award and this really lovely medal. The award recognizes many years of mRNA research and translation of mRNA technology into the development of a vaccine to combat a pandemic. I share this recognition with my esteemed colleagues and friends, with Ugur, with Katie and Drew, and the multiple generations of scientists and researchers who have contributed to understanding mRNA, improving it, and learning how to apply it. As with any breakthrough innovation, it is the confluence of many efforts that has allowed the practical use of mRNA technology as a powerful tool in the service of humanity. I also want to thank our incredible team at BioNTech, and our partners who have contributed. This award is particularly special for me because it comes from the Cancer Research Institute, an institution with long tradition that represents the community of cancer immunologists, the very community in which I grew up, in which I am deeply rooted, and which I consider my scientific home. So thank you again.
Congratulations, Dr. Türeci, and to all our honorees this year for their outstanding achievements and life-saving work. Earlier, the Cancer Research Institute had an opportunity to sit virtually with Drs. Karikó, Sahin, Türeci, and Weissman for an exclusive roundtable discussion about the future of cancer treatment and the role their mRNA vaccine technology will play in shaping that future. The impact of their work stands to spark a second immunotherapy revolution, not only in the treatment of cancer, but also in treating myriad other diseases. We now invite you to join us in taking this future look to learn what lies on the near horizon in cancer treatment. Award-winning health and medicine reporter Alice Park of Time Magazine moderates this exciting and inspirational discussion.
Congratulations to all of the winners of the 2021 William B. Coley Award from the Cancer Research Institute. I'm Alice Park with Time, and it's my pleasure to be moderating this discussion about probably the most influential product in the past year, the mRNA COVID-19 vaccine. We have with us today some of the key leaders involved in developing this vaccine. I think all of us would agree that we're in a very different place in 2021 than we were in 2020. And that's largely due to the work of the 4 scientists we'll be hearing from today. Welcome, everybody, and let's dive right in. Last year, we saw a vaccine developed from scratch in less than a year. It went from nothing to getting into arms, authorizing getting into arms in less than a year. That's lightning fast for any medical treatment, let alone a vaccine. But that speed, I think, really disguised the years of work that had already been done to lay the groundwork for making it possible to develop a vaccine in such a short time frame. So Drs. Karikó and Weissman, I'd like to start with you. What convinced you that mRNA technology had promise? And I believe you had a hard time convincing others throughout your career about your belief as well. So let's start there with what was it about mRNA that gave you such confidence and that made you think that this was something that really would begin to help us in the future?
So in the early 90s, I started to work with messenger RNA and try to develop for therapy. I met Drew, who was interested to develop messenger RNA, or he was interested to develop HIV vaccine. And when I mentioned that I am working with messenger RNA, he got interested to make vaccine with mRNA. And this is how we started.
That was almost 25 years ago. And people have to understand there was an enormous amount of work. There have been many human clinical trials with RNA vaccines. So this isn't new technology. I think what happened this year is the technology was poised to be used, and that's why it made a vaccine so quickly.
And I'd also like to hear from Drs. Sahin and Türeci as well. When you founded your company, BioNTech, your focus was on finding a vaccine for cancer. And you had your choice of technologies to choose from. Why mRNA? What was it about mRNA that convinced you and that made it stand out in your minds?
We started with mRNA before we founded our company, also in the 1990s, and with the intention to develop a cancer immunotherapy, a cancer vaccine. And as the friends and family of the CRI might remember, in those days, different technologies were tried out, peptides, proteins, DNA, and so on, for cancer vaccination. And we were attracted by mRNA because it was so versatile. It brought its own adjuvant activity, its intrinsic adjuvant activity, and it was also, in terms of production, in terms of manufacturing, it had a high potential. And we were at that time interested in personalized cancer vaccines, which we thought mRNA would be ideal for. And that meant, however, that a couple of disadvantages of mRNA had to be addressed, and that meant many years of work.
I think what the story of Katie, Drew, and our story is about is that we started with different intentions, came to the same conclusion that mRNA might be useful, and were encountering the same challenges at the beginning because we wanted to use it, but it was not ready for use. So we and other groups started to work to solve these problems.
Let's talk about some of those problems with mRNA, because you've alluded to, all of you, that it wasn't quite ready. And I know from reading some of the literature around that time that one of the real pitfalls of mRNA was its instability, compared to DNA. Can you talk a little bit about those challenges and why, despite them, all of you still felt that this was a avenue of research worth pursuing?
I mean, I might mention that the messenger RNA is labeled in the body and on the shelf. Also, most of the laboratory they work with plasmid, they use the RNAs to purify the plasmid in molecular biology lab. And most of the laboratory were contaminated with RNAs. I never used RNAs in my lab. And so that is why, the RNA was okay. And I did not have that much of a problem. It's more problem was related that when Drew showed that the messenger RNA, conventional one, was inflammatory, because my major goal was to develop for therapy. And I was worried that if try to use the messenger RNA for treating patients and who already had problems, then we will worsen their situation. And so inflammation caused by the conventional mRNA was a major problem for me, at least, or for us to solve.
Yes, for us it was an intended effect. So we were amazed by the opportunity that mRNA itself could act as an adjuvant. But we also realized that there is a discrepancy between the amount of intended adjuvant activity and the amount of intended protein expression. So we tried to make the mRNA more potent with regard to the protein expression to get more or less a perfect mix of both functions.
And Dr. Weissman, I wanted to ask you about your work, as you said, in HIV, where you began with this, because it's interesting, I think, when we look at this mRNA vaccine, that we've really seen sort of the intermingling between work that was being done in cancer and immuno approaches toward targeting tumors, and work being done in infectious diseases to target viruses. Now, obviously, a tumor and a virus are very different animals, but they do share some things. Can you talk a little bit about the work you had been doing in HIV when you had met Dr. Karikó, and how collaborating with her really moved your work forward as well?
So I worked in Tony Fauci's lab during my fellowship, and we studied HIV immunology. And I developed the study of dendritic cells. Those are the cells that start all immune reactions. So when I came to Penn, I wanted to work on vaccines. And dendritic cells are the principal cell that starts vaccine reactions. So I was looking for ways of loading dendritic cells with the antigen of interest. And my principal interest was for HIV. So when Katie and I met, it seemed like the perfect meeting. I joke, it's like the Reese's commercial of peanut butter and chocolate coming together. And then we started working together, and it was many years before we actually worked on an HIV vaccine, just trying to understand RNA, inflammation, and everything else. But now we have very active programs. We have 2 clinical trials with HIV vaccines that will start early next year. And we're hoping to have many more.
And for all of you, you've alluded to this ability of mRNA to act as an adjuvant or really kind of be a jumpstart for the immune system. And obviously, when we look at COVID-19, that was a very key feature. Drs. Sahin and Türeci, when COVID-19 hit in 2019, 2020, what made you think immediately that, oh, our work in cancer could help in this area? Again, two very different areas, cancer and infectious diseases. What made you think that the work that you had been doing might be useful against COVID-19?
Those areas are actually not so different. It's about delivering with the mRNA the antigen, the immunogen against which the vaccinee's body is supposed to induce an immune response. And we had, as we already pointed out, worked on this in cancer and had improved our mRNA platform such that it could actually go into clinical trials in 2012 already. And until the pandemic hit, we had already treated in several clinical trials more than 400 cancer patients, also with personalized vaccines, where we had to ensure very short turnaround times between analyzing the patient's cancer and on-demand manufacturing an mRNA vaccine for that, so that the clinical development part, some safety data, and the fast manufacturing were already in place. And it was about now adapting the platform, which had already entered clinic and had been vetted in clinical development, towards virus-directed vaccine. So our technology at that point was ripe, which was good. 1 year earlier, it would have not been the case, and we could then pivot towards the COVID-19 vaccine development.
And Dr. Sahin, I know that when you saw the first cases being reported of COVID-19, you had the same thought, that the work you had been doing, the groundwork that you and your team had laid could be very applicable to this new pandemic. Can you talk a little bit about that moment when you realized that, the work you had done in cancer could be very, very useful against this infectious disease?
Yeah, so we had on January 24th, there was an online publication in Lancet describing infections in Wuhan, the outbreak in Wuhan. And based on this publication, we came to the conclusion that this is not only a local outbreak, but this is going to become a global pandemic. And as Özlem said, we knew that we have a technology in place which could make fast vaccines, but we also knew that we don't have experience with infectious disease vaccines. Yeah, so we had experience with preclinical work, but the situation in human is always different. You can't take the data from the preclinical and just expect that the same outcome will happen. Therefore, we started with more than 20 candidates to be able to come up with an optimal balance between antigen delivery and immune stimulation. And in our preclinical results, it was not obvious which of the mRNAs will make it. But in the clinical setting, we realized that the nucleoside modified mRNA, based on the discovery that Drew and Katie made, in combination with the lipid nanoparticle that we're using in this clinical trial, was the best one. It turned out in the studies that the full spike protein has some advantage with regard to safety, which was also not expected, and has advantage with regard of the T-cell immunity. We have two parts of the immune system. One part is the generation of antibody responses. It is believed that the antibody responses prevent the infection. But once the virus has entered the cell, we need the T cells. And we were able to see both types of immune responses, particularly also strong CD8 T cell responses. And therefore we were confident that the vaccine in principle is suitable, to be active. But there was another unknown. The unknown was that this group of viruses, the coronavirus, there was no evidence before that such vaccines in humans will work. The whole family of coronaviruses, there was no pre-evidence for any type of vaccine, not only for mRNA. So we were of course knowing that the pharmacodynamic activity of the vaccine is good, but we did not know if the vaccine will work until we got the data, the phase 3 clinical data. This was on November 8th, and we learned that we have a vaccine efficacy of more than 90%, which was, of course, a wonderful feeling, because at that time point, we not only understood that we have a vaccine, but that the world has a vaccine, which can benefit to control, which can help to control this pandemic.
I think you're being very modest when you talk about those results, of over 90% efficacy, because as you said, there was never any demonstrated effective vaccine against any coronavirus. There had been no authorized or approved vaccine using mRNA technology. So I think, we can't gloss over the huge advance that was. I'd love to hear from all of you who had such critical contributions to this vaccine about what that moment was like to realize that the investments you had made and the decisions you had made to really bank on mRNA technology had come to fruition from all of you. I think that would be really useful for us to hear, because this is really a huge milestone.
I worked with Drew from '97, and the last 8 years I am at BioNTech. So everybody here is my colleague, and I had fun to work with Drew and getting from the start and going through a lot of struggle, but we worked very hard. And then that happened also when I was at BioNTech, and we worked very hard to improve the RNA. And then all of the colleagues there and all of our colleagues at Penn and also at Pfizer, everybody, that just it culminated that all of these people, in addition to those who we relied on their research, who did decades ago, people who are not with us anymore, all of those people contributed. And it was somehow that the realization that in my lifetime, I can see the benefits of our work. And it was I was very late. I was humbled. I was, feeling all of this emotion that, that our work ended here. And I really, not take personally the whole thing. I am just considering myself representing all of those scientists who worked for many, many years, and in anonymity. And I was one of those, a year ago. And so I am very happy that so many people are helped.
I completely agree. The big worry was, of course, the phase 3 results. And when we heard those, it was just incredible excitement, because no vaccine, no respiratory vaccine had efficacy that high. We had been working on vaccines for 10 years, and just about every vaccine we made had 100% efficacy in animal models. But that still left a lot of worry, because humans are always different than animal models. Everything works in mice. Things rarely work in humans. So it was a combination of relief and just incredible satisfaction and joy, because my dream since being a clinician scientist was always to help people. And this vaccine helps people.
As my colleagues already pointed out, it was a very special moment. The entire team which hands-on worked on this vaccine had basically worked 24/7. So everything before this readout was like a blur. And on this Sunday, we were sitting at home and waiting for the data. And at that point, it was not clear how the data would look like, whether the thumb would be up or down. Ugur actually said to me, irrespective of what we will hear later today, it was our obligation to contribute, and we did our best. And in fact, we had put everything which had been developed over the last 2 decades into this vaccine, all design elements of improvement, and more was simply not possible. And the call which we then received directly after the independent data review board had seen the data was relief, and felt like a blessing to hear that we had a vaccine which worked, but also that it was 95% efficacious, which really translated into everyone who will be vaccinated has a very high probability to be protected. That was simply an amazing moment.
Yeah, I agree with everything what the colleagues said, and I had very similar feelings. The greatest feeling that I had was I was grateful. I was really grateful that nature turned out to be
merciful.
and I reflected all the things that came together to make that possible. Yeah. So one of our friends said that the stars aligned in a manner, in a manner that the history was that this historic event was possible. So, and I felt just this so grateful that we had the chance, as Drew said, as scientists, to make a difference. Yeah, that is what we all want to do, but being able to do that even at that scale, it is just a blessing.
And I want to go back now then to look at how the lessons that we've learned from that experience, that whirlwind year, translating that technology to a COVID-19 vaccine, how that's now going to help in the continued work to perfect a cancer vaccine. I want to just get some more details about the work Drs. Karikó and Weissman, you did to help with what Dr. Türeci talked about, the important delivery of the antigen. Can you talk a little bit about how much of that was in the vaccines that were already being tested in the few hundred people in cancer, and whether the work in COVID and this past year helped to accelerate some of that or perfect some of that?
Well, we started begging for lipid nanoparticles the first time they were published. And it took us nearly 10 years to finally get them. And that was 10 years of constant pestering of everybody who had them. But when we finally got them, it changed the vaccine world in our minds. And just a note, I apologize for the science, but the cancer vaccine and the COVID-19 vaccine are different. The cancer vaccine uses an unmodified RNA that supplies the adjuvant activity. The COVID vaccine uses a modified and purified RNA that doesn't have adjuvant activity. And what that allows is the lipid nanoparticle supplies a very special adjuvant activity that's different from any other adjuvant. And that adjuvant activity, in my mind, is what makes the vaccine work so well and be so efficacious. So we wanted lipid nanoparticles for years. We finally got them. They turn out to be a really critical component of the vaccine. The delivery vehicle isn't simply a way of protecting RNA. It also supplies the adjuvant that makes the vaccine work very well.
And Doctors Sahin and Türeci, I wanted to ask you about those trials in the several hundred cancer patients. Can you bring us up to speed on what you were seeing? I know they were just beginning, but what kind of results were you seeing, and how is the work now, the results that we're seeing from the tens of thousands, millions of people who received the COVID vaccine going to inform those trials, those cancer trials, as they move forward?
The results we are seeing in the cancer trials is that on the one hand, safety, we see an excellent safety profile in the cancer patients. We see pharmacodynamic activity, which means that we can induce, in particular, CD8 and CD4 T cell responses in cancer. With antigens being intracellular, the T cell responses are the critical ones. And what we have achieved by the design elements, which we also use in the COVID-19 vaccine, is that the induction of the T cell immune response is highly potent. We have cancer patients who have 10% of their peripheral T cells directed against vaccine antigens. These are magnitudes you would normally want to see with adoptive T cell transfer. So the activity is very high, and the COVID-19 data, which uses this design elements as well in order to get high potency, confirms further that these design elements are potent, and we will continue to use them in cancer. And what we also see in our cancer trials is we have shrinkage of tumor of metastatic lesions. In cancer, as you know, we are not preventing with a vaccine, we are treating an already existent tumor burden, and our T cell responses we generate are able to shrink tumors. We have objective responses in cancer patients, and this motivated us actually to use the same design elements of the mRNA, the stabilizing and potency increasing design elements also in the COVID-19 vaccine, where this has been now validated.
I think what is really important is, as Drew mentioned, there are different mRNA platforms, and there will be more in future. So in the cancer field, we really need extremely strong T cell responses, and what we have accomplished in the last 7 years in clinical trials is showing that with the mRNA vaccine that we had developed, which is targeting after intravenous delivery into dendritic cells, we get extraordinarily strong T cell responses, and this T cell responses translate into a clinical activity. We have to show now in randomized clinical trials that this clinical activity makes really a difference with regard to the outcome, survival of the patients. So these are running clinical trials, and we will get the next set of data next year. Yeah, but what is really encouraging is that the diversification of the mRNA field. We have this nucleoside modified mRNA, which would have most likely a wonderful future, particularly for delivering proteins that should not be immunogenic. We have the vaccine field, cancer vaccine field, infectious disease field, where different type of mRNAs might be suitable. There are different tools for delivery, lipid nanoparticles for IM delivery, lipid nanoparticles for IV delivery, polyplex particles, and so this is a multitude technology toolbox which will be useful to generate really hundreds of applications, and that is the beautiful future that we are seeing, and it is just the beginning. Yeah. And it is, of course, a fantastic beginning because usually technologies start with a small indication, and now the first mRNA mRNA medicine just became a medicine which is saving humanity from this pandemic.
Do you think that the flexibility of the mRNA platform that you just alluded to there, would that apply to being able to have more precise and I guess specific approaches designed for different types of cancer? Will we likely see a different one type of mRNA approach against like solid tumors, for example, versus blood cancers?
Cancer is indeed a very different beast, and every cancer is different. Every type of cancer is different. So we are, as Özlem said, dealing with existing tumors, and even a small existing tumor in humans consists of about 1 billion tumor cells, and large tumors have hundreds of billions of tumor cells. So what we really need to address in cancer, not only to induce immune responses, but ensure that the T cells can go into the tumor, affect the tumor microenvironment, are able to fight and kill tumor cells faster than new tumor cells are growing. Yeah? And that would mean, for example, combination therapies. We are using, at the moment in clinical trials, it's an early phase, we are using an mRNA delivering an IL-2 molecule. Yeah? So you know IL-2 is one of the first immune modulators in cancer therapy, and one of the big disadvantages is the pharmacokinetics with a high toxicity profile because it has to be dosed at a high level. By delivering with mRNA, we manage a much better pharmacokinetics with a lower toxicity, but still pharmacodynamic activity. So the field of cancer therapy will not only be vaccines, but also delivery of cytokines, delivery of antibodies. Particularly the delivery of antibodies will benefit from the nucleoside modified mRNA technology because the amounts of mRNA which are delivered are very high, yeah, and here the immune stimulatory mRNA is just not tolerated at that level. So we will see combination therapies, not only with mRNAs but also other type of existing treatments, for example, checkpoint blockade.
And I want to go back to Drs. Karikó and Weissman for a minute too, and get a perspective from you on every success always generates more questions, right? And especially in the research world. I'm curious about where you see your research leading next.
We have a lot of exciting things that we're working on. We're working on a bunch of different vaccines for things like malaria, hepatitis C, genital herpes, tuberculosis, and many others. The most exciting things that we're working on are gene therapy, which is something new for mRNA. But nowadays, so many gene therapy systems, things like CRISPR-Cas9, are delivered with mRNA. And we figured out how to target those therapies in vivo. So the hope in the future is that we can do gene therapy treatments with in vivo administration. And as an example of why that could be important, 200,000 people a year are born with sickle cell anemia in sub-Saharan Africa. You can't treat those with bone marrow biopsy and an infection with a virus. But if we can figure out how to do an in vivo gene therapy, where we give them a single intravenous dose of mRNA LNPs that fixes the genetic defect, that's groundbreaking to me. And that allows us to treat difficult diseases all over the world with very simple means.
And Dr. Sahin and Türeci, I'd love to hear from you as well. And 5 years down the road, let's say, where do you hope the mRNA technology platform will bring us when we look at both the research and clinical opportunities that this experience has now given us. Where do you envision this platform will be in 5 to 10 years? I think 5 years ago, it was very different. I don't know if anyone would have envisioned we'd be here today, but still, let's take that crystal ball out and see and hear about what your perspective might be on where we might be in 5 years with mRNA technology.
Actually, we wrote a paper 7 years ago together with Katie, Özlem, and I, being extremely optimistic in a review about what is going to happen in the next years. And when we submitted this paper, it is published in Nature Reviews, Nature
Drug
Discovery. When we submitted the first version of the paper, the editor commented that this was too optimistic, and they want to get a more realistic view of that. You remember, Katie, the feedback from the editor. And actually, I had a look into the optimistic version that we had to revise. And it was completely what we had predicted in this paper. That we expect that mRNA technology is validated. And that we are going to see applications in cancer and infectious disease to be successful, and at that time already the paper described multiple applications from different groups. A number of groups. The mRNA research community is not the biggest research community, but it is a very consistent and persistent research community. The community had already shown the use of mRNA for allergies, for autoimmunity, for regeneration, for reprogramming of cells. So there are, I think, it is difficult to find a medical application which cannot be addressed by mRNA.
I agree with Ugur. I also believe that mRNA has a broad transformational potential. I'm not sure whether it will be actualized within the next 5 years, but for sure within the next 10 years. And we like to compare it with what happened with recombinant DNA technology, which 40 years ago meant that you would not need to extract insulin to treat people from animal organ tissues, but could put a piece of DNA into a cultured cell and get the insulin from there. And that revolutionized an entire industry. And now with mRNA, you don't need the sort of middleman of a cultured cell. You can let the patient become their own biofermenter, so to say. And I think this really has the potential, together with a broad panel of indications where mRNA can be used, this has the potential to transform.
Well, I want to thank all of you for this fascinating discussion and for your pivotal contributions to making it possible for us to really start thinking about controlling rather than succumbing to this pandemic, and thinking about novel new ways of controlling cancer as well. We're in a very different place this year than we were last year, thanks to your work. And it's clear that we're just at the beginning of the promise of the mRNA platform. Thank you to the Cancer Research Institute for organizing this discussion. Congratulations again to our honorees, and we all look forward to hearing more about your work in coming years.
Thank you, Alice. This was a great pleasure.
Thank you, Alice, for moderating that fascinating and insightful discussion. It not only gave us a rare opportunity to meet the scientist whose work is preventing the deaths of millions of people right now, but it also demonstrates the vital importance of scientific research that makes possible the life-saving medical advances of today and the near future. On behalf of the Cancer Research Institute and the CRI Scientific Advisory Council, I once again congratulate our 4 honorees, Drs. Karikó, Sahin, Türeci, and Weissman, for their outstanding achievements. And as we conclude this 35th annual awards ceremony of the Cancer Research Institute, I'd like to thank all of those whose generous support powers the work of this dynamic and visionary nonprofit organization. You are helping to make possible a truly exciting and hopeful future immune to cancer. Thank you.